Hello everyone, welcome to the channel Microbial World. In this video, we will be learning about type 4 hypersensitivity reaction in detail. Type 4 hypersensitivity is a cell-mediated immune response that occurs 24 to 72 hours after exposure to the antigen. As the immune response is taking time to develop after antigen exposure, it is also known as delayed type hypersensitivity. Unlike other hypersensitivity reactions such as type 1, 2 and 3 hypersensitivities which involves antibodies, type 4 reactions are mediated by T cells and macrophages. The mechanism of type 4 hypersensitivity can be classified into two phases: sensitization phase and effector phase. Sensitization phase is the phase where the immune system is first exposed to the antigen, and effector phase is the phase of second exposure to antigen and inflammation. Sensitization phase. In the sensitization phase, when an individual is first exposed to the antigen, the immune system recognizes the antigen and generates memory T cells. The first step involved in the sensitization phase is antigen processing and presentation. An antigen is entering into our body. The antigen could be tuberculin, metals, poison ivy, or any bacterial antigen. When the antigen enters into our body, antigen-presenting cells such as dendritic cells, macrophages, or Langerhans cells in the skin will capture the antigen and process the antigen. It will present the antigen to naive CD4 T cells via MHC class II molecule in the lymph nodes. The interaction between MHC class II molecule and T cell receptor activates naive CD4 T cells, leading to their differentiation into T helper 1 or T helper 17 subsets. It depends upon the cytokine environment. Th1 differentiation is promoted by interleukin 12 that is secreted by macrophages and dendritic cells. Th17 differentiation is promoted by interleukin 6 and TGF-beta that is transforming growth factor beta. Activated Th1 and Th17 cells will proliferate and differentiate into memory T cells which remain in circulation and lymphoid tissues. These memory T cells will remember the antigen and react more aggressively upon future exposure. Effector phase. When the same antigen enters the body again, it triggers a strong inflammatory response as the memory T cells quickly recognize the antigen and react to it. So in this phase, antigen-presenting cells such as macrophages or dendritic cells will present the antigen to memory T cells in the tissue. T helper 1 cell and T helper 17 cells will quickly recognize the antigen and become reactivated. Activated T helper 1 cells release interferon gamma. Interferon gamma activates macrophages, making them more aggressive. Activated macrophages secrete pro-inflammatory cytokines such as tumor necrosis factor alpha, interleukin 1 and interleukin 6 which promotes local tissue inflammation. They also produce reactive oxygen species and nitric oxide, leading to oxidative stress and tissue destruction. Proteolytic enzymes are also secreted by macrophages, breaking down the extracellular matrix and contributing to tissue damage. Whereas activated T helper 17 cells will release interleukin 17. Interleukin 17 recruits neutrophils and monocytes to the site, amplifying inflammation. Recruited monocytes will differentiate into macrophages and increase the production of pro-inflammatory cytokines such as interleukin 1 and tumor necrosis factor alpha. So the accumulation of macrophages at the site leads to chronic inflammation and granuloma formation. Pro-inflammatory cytokines such as tumor necrosis factor alpha, interleukin 1 and interleukin 6 along with reactive oxygen species, nitric oxide and lysosomal enzymes collectively contribute to tissue damage. Examples of type 4 hypersensitivity reaction. Tuberculin skin test. In this test, a reaction to Mycobacterium tuberculosis antigens that is injected into the skin causes redness and swelling after 48 to 72 hours if the person has been previously exposed. Another example is contact dermatitis. This is a skin inflammation that is caused due to contact with allergens like poison ivy, nickel, latex, or cosmetics, which is triggered by a T-cell mediated immune response. Granulomatous inflammation causes chronic infections like tuberculosis and leprosy, leading to macrophage activation and granuloma formation. Graft rejection is another example of type 4 hypersensitivity reaction. It occurs in organ transplantation where the T cells attack the transplanted tissue weeks to months after transplantation. Autoimmune diseases. Conditions like type 1 diabetes mellitus, multiple sclerosis, and rheumatoid arthritis involve T-cell mediated destruction of self-tissues. So this is all about type 4 hypersensitivity reactions. Thank you for watching.